from any that you've heard earlier in the day. First of all, I'm not going to give the whole lecture because I have trouble with my voice under pressure. I decided I'd give the first part that pertains to Larry, and then Bonnie, who looks a lot like me, actually, will give the second half. We're phenotypically very closely related. Anyway, in 1965, I was one of 11 first-year students to enter in the Department of Genetics here at the University of Washington. Because of my background, I graduated from the University of British Columbia. I felt a little intimidated to be surrounded by students from Ivy League and other highly rated schools, Harvard, Amherst, Brown, Oberlin, Reed College, Brooklyn College, to name just a few. But I needn't have worried, because I soon found that being in the department was like being part of a closely knit family. And the fact that the students and faculty were on a first-name basis helped remove barriers between us. Among the incoming students, 3 of us, Brian Davis, Lenny Robbins, and myself, wanted to go into Larry's lab because we'd all had previous experience in Drosophila. Larry had little say in the matter because, in fact, he wasn't there. Rather, he was in Rome, Italy with Dan Lindsley, searching for meiotic mutants in a wild Drosophila population. So Brian, Lenny, and I went to Herschel Roman's office together, and we told him that we wanted to be in Larry's lab. And I'll never forget what Herschel said next. And he chuckled as he said it. Well, the golden age of Drosophila genetics is past, but if you want to be in Larry's lab, that's fine with me. Herschel was a very wise man in so many ways, but he missed the boat on this one. Just think of how delighted he would have been to know that 52 years later, Jeff Hall, one of Larry's graduate students, would be co-winner of the 2017 Nobel Prize in Physiology or Medicine for his work in Drosophila, elucidating a genetic basis of circadian rhythms. What do I remember about Larry? He was imposing physically, intellectually, and in his commitment to being fully present in every moment of his life. There was nothing tentative about him. To say that he had a larger-than-life personality would be an understatement. I soon learned that Larry's office door was always opened to us and any other students or faculty members in the department. In addition to Brian and Lenny, the other members of our group at that time included Bruce Baker, Adelaide Carpenter, Jeff Hall, and Stephen Miller, who joined us between 1966 and 1969. Larry invited us to have lunch with him in his office, which we did almost every day. Larry would regale us with anecdotes about his childhood growing up in Brooklyn. We learned that as a young boy, he was festooned with the garland of garlic that was intended to keep evil spirits away, and perhaps more remarkably, that he attended but did not graduate from Lafayette High School, a place that he did not enjoy. Fortunately for him and for us, he found his path to higher education through the Cornell Agricultural College, which admitted students lacking a high school diploma. That Larry grew up in the concrete environs of Brooklyn probably accounted for his lack of interest in the great outdoors at that time in his life. And this would explain why the lawn surrounding his Seattle home consisted of astroturf, which I did not believe until I saw it with my own eyes. And that after driving his father to see Mount Rainier, Larry told us that he had actually rolled down the window of his pink Oldsmobile so that his father could see the mountain better. Lunchtime discussions covered a wide range of other subjects, including Italian politics, history, family, food, and more. As has been mentioned earlier, we also gathered in Larry's office to analyze classic papers in Drosophila genetics under his guidance, so that we might begin to understand the principles guiding the genetic manipulation and behavior of chromosomes, what he called chromosome mechanics. We also learned the history of Drosophila genetics, who the key players were, and where we were on the pedigree of Drosophila geneticists. Separate from imparting knowledge of genetics to us, Larry was very interested in and concerned about current events and activities on a global scale. The lab was a setting for lively discussions about events that impacted our lives, most prominently during my years here, the Vietnam War. Several of my lab colleagues were draft eligible and indeed actually reported for their draft physicals. Fortunately, all were declared 4F for reasons: a weak ankle, difficult to correct vision, that made them unfit to serve in the Army, but had absolutely no impact on their ability to score fruit flies and otherwise function well in Larry's lab. Larry was a doting husband and father. He frequently mentioned Iris, Jack, and Dee to us, and clearly took great delight in their presence in his life. I think because family was so important to him, he wanted to extend the idea of being part of a family to the lab. This desire contributed to the lab's informal atmosphere and Larry's relaxed interactions with us there. The concept of family extended to family units. When Brian's son Christopher was born, and then Lenny's son Daniel arrived, Larry strolled down to the nursery in the UW hospital to see the infants and then congratulated their happy mothers, Jan and Diane. Later, both children were frequent visitors to the lab. Adding to the sense of family was the tradition established by Avril, Larry's dedicated technician, of celebrating every student's birthday by baking a special cake for the occasion. For me, the cake always included poppy seeds, and I still have her recipes. Brian, Lenny, and I were each given a meiotic mutant captured from the Italian countryside to investigate as our thesis project. Mine was my S282. Based on a very small data set, Larry had an hypothesis about how this mutant disrupted recombination and chromosome segregation during meiosis. It was my responsibility to devise and carry out sequences of crosses between it and other strains of Drosophila, count hundreds if not thousands of progeny, and analyze the data to find out whether Larry's hypothesis was true. Before our final thesis defense, each of us gave an informal presentation to the lab about our results. After my presentation, Larry said nothing in front of our group, but later he came over to me and said, "Whatever happened to the perfectly good hypothesis that I gave you?" Well, as you can imagine, I just about died. I immediately thought, what a disaster. What am I going to do now? Well, I decided to turn to Brian, whom I considered to be the most mature student among our group, and the least likely to think less of me if I had made a big mistake. I asked him to review my data and the analyses with me. We sat down and looked over the data, and fortunately, the results were exactly as I had presented them. Buoyed by this, and with somewhat more confidence than before, I approached Larry and asked him if I could go over the data with him. Of course, he said yes, so I presented the results and my interpretation of them. After a few minutes, Larry said, "Oh, now I see." The relief that I felt was enormous. Looking back, I realized that this episode held both the lowest and highest points of my graduate education. I had passed the test presented to me by nature and unexpectedly demonstrated to Larry that I had also learned what he had taught me, to apply my knowledge of Drosophila genetics to carrying out informative crosses and to interpret the results with an open mind. Needless to say, both Larry and I were very pleased with the outcome. In thinking about my years as a graduate student and preparing for this event, I realized that I was very fortunate, because even at the time I was in Seattle, I knew that I was enjoying a very profound and worthwhile experience. I recognized that my graduate school years were very good years, and I even suspected that they might be a high point in my life, and I didn't have to wait until years later to fully appreciate them. But as much as I enjoyed my graduate student experience, I knew that I did not want to run a Drosophila lab, and I did not see myself being a Drosophila geneticist. In fact, I had no clear-cut plans for my future. I think that Larry accepted my rather nebulous state of mind and hoped for some good outcome for me. Should I try to keep on going? Yeah, I know. As long as I don't spill it on anything important. Like the computer. I may have the distinction of being the only person to have been both a graduate student and a postdoctoral fellow with Larry. Herschel and then Larry welcomed me back to the genetics department after my postdoctoral fellowship elsewhere at the university ended early. And that extra time in the genetics department changed my life. While back in Larry's lab, I shared office space with David Cox, and Wendy Raskind was a frequent visitor. Both were in the MD-PhD program, and their research was based in Stan Gartler's lab, whose office was just across the hall. Various aspects of medical genetics were frequent topics of conversation, which I found very interesting. And soon enough, I was attending the medical genetics post-clinic conferences run by Arno Maltowski, adult diseases, and David Smith, pediatrics, particularly dysmorphology. Within a short period of time, I knew that medical genetics was the field that I wanted to be in. But how was I going to get there? Another University of Washington colleague, Gil Oman, provided the bridge to that goal. He referred me to Fred Bergman, chief of the extramural genetics program in NIGMS at NIH in Bethesda, Maryland. Fred had a project that required someone to review medical genetics record-keeping systems at clinics throughout the US and Canada and assess their utility for research use. The project lasted about 1.5 years and provided me with the wonderful opportunity to meet outstanding medical geneticists and learn about their clinics and training programs. The intramural NIH program was quite small at that time, and it was easy to meet people with common interests. In 1976, I was hired as a fellow by Robert Miller, chief of the clinical epidemiology branch in the Division of Cancer Etiology in the National Cancer Institute. The focus of the branch was to conduct research to identify and study familial cancers and cancer syndromes. Right up my alley. I stayed at NCI through various permutations of the branch and the division until I retired in 2007. I'm still affiliated with the program as a special volunteer. In the late 1970s, I was one of a group of 14 investigators from a number of different intramural NIH institutes who got together to discuss forming a medical genetics program at NIH. I was the most junior member of the group and delighted to take on the tasks of organizing trans-institute activities for us to participate in. We began with a journal club, then branched out to giving presentations of our clinical research, which later became enhanced with the presentations of relevant patients. Within a few years, we obtained dedicated space 1 day a week for a genetics clinic, invited fellows who were physicians already doing research in our labs or branches to rotate through the clinic, and were given a permanent position for a genetic counselor to run the clinic by the director of the clinical center, which is the research hospital at NIH. Finally, in 1980, the deputy director of NIH committed 3 full-time fellowship positions for physicians to the program. Like the genetic counselor, these were attached to the clinical center rather than any one institute. Over time, our weekly genetics clinic was followed by a post-clinic conference We developed didactic courses in various aspects of medical genetics and established rotations through the genetics clinics of neighboring institutions. In addition to their clinical training, the fellows also participated in research in labs and branches throughout NIH. The NIH Interinstitute Medical Genetics Program was accredited by the American Board of Medical Genetics in 1982. I was credentialed as a PhD medical geneticist in 1984 and developed a clinical program, research program, whose participants were seen in the clinic by me, my co-investigators, and the program's fellows. The training program was run by a steering committee that comprised clinician investigators from various institutes who participated in the program. I held a variety of positions on the steering committee over many years, including co-director, associate director, and director. I remained on the steering committee until 2005. Being involved in the development of the Interinstitute Medical Genetics training program and participating in it were undoubtedly the most fulfilling aspects of my career. I was part of a small group of highly committed clinical investigators who created a dynamic, broad-based, highly collegial interinstitute and interdisciplinary training program in medical genetics that offered me opportunities to conduct protocol-based clinical research, mentor outstanding fellows in clinical activities, develop and teach in didactic courses, and develop leadership skills. In retrospect, the program we created mirrored all the best aspects of what I had observed and experienced during my years in Larry's lab and in the Department of Genetics here at the University of Washington. So, what type of clinical research does a former fruit fly geneticist do? All my clinical research has had the same goals: to identify genes and the mutations in them associated with a disease, to delineate the clinical phenotypes associated with specific mutations, to educate patients about the genetic basis and natural history of the disease, and to offer patients and their at-risk relatives genetic testing or other types of screening to aid in early detection and treatment. I have been involved primarily in 3 major clinical research programs, but today I'll describe just one of them. By chance, all 3 have focused on individuals with an increased genetic risk for tumors of the neural axis, including the brain through the spinal column down to the tailbone. Our most recent research endeavor has been elucidating the genetic and clinical aspects of familial chordoma. Chordoma comprises about 1 to 4% of all primary bone cancers, 30% of spinal cancers. It is rare, with an incidence rate of less than 1 per million persons per year. It is more frequent in males than females and in Caucasians than African Americans. Its incidence rate increases with age and is rare in people under the age of 40 years old. The median age at diagnosis is about 58 years. Chordomas of the skull base are somewhat more frequent than chordomas of the spine and sacrum, and predominate among younger individuals, including children and females. About 30% of chordomas present in the skull base, 29% in the spine, and 31% in the sacrum or at other sites. Chordomas are believed to arise from remnants of the notochord. The notochord is the main axial structure in vertebrate embryos. It provides physical support, patterns the central nervous system, and ensures proper development of a variety of organs. The notochord regresses as ossification of the spine proceeds in late fetal life and vertebral bodies form, and its remnants become the substance of the intervertebral discs. In addition, some ectopic notochord remnants persist in vertebrae and in bones at the base of the skull into adulthood. It is from these ectopic remnants that chordomas are believed to develop. Chordomas are slow-growing but locally aggressive and invasive. Although in the past, some patients were told that chordomas are benign, they are in fact malignant and can metastasize. Because chordomas are mostly resistant to conventional chemotherapies, the main treatment is surgery, followed by radiotherapy given as an adjuvant. Traditional chemotherapy can have a role in locally advanced or metastatic disease, but there is a need for more effective treatment for tumors that recur or spread. Most chordomas are sporadic, but to date, 10 families with at least 2 blood relatives with histopathologically confirmed chordomas have been described worldwide. Some of these families have formed the basis of our research. My interest in chordoma began when this large multiple case family was referred to our branch. Our clinical approach to chordoma families has been to use neuroaxis MRIs to screen individuals with chordoma as well as selected first and second degree relatives. This family was referred with 4 individuals with chordoma: this man, his sister, a niece, and a cousin. We identified through screening 4 additional cases: the son of this man, his father, his aunt, and one other of her sons. And then there's 2 additional cases that we believe have chordoma, but the lesions have not been excised or biopsied or excised. Over time, we ascertained 6 additional families, but did not identify any new chordoma patients in any of them. Altogether, we had DNA and clinical data on 23 chordoma patients and over 40 relatives from these families. We began with gene mapping studies. Using a genome-wide linkage scan followed by fine mapping with microsatellite markers, we found significant evidence for linkage at 6q27. The minimal disease region was 11.6 megabases and contained 74 genes. The most biologically plausible gene was the T gene, also called brachyury. Brachyury is a tissue-specific transcription factor that is essential for proper notochord development and maintenance. It is specifically expressed in the chordomas and the notochord, but not in a wide variety of non-neoplastic tissues or over 40 other types of tumors, including chondrosarcoma, with which chordomas are frequently confused. We sequenced the coding region and adjacent spike sites of the T gene and upstream and downstream of the coding region, but did not find any sequence variants consistent with a disease-causing mutation. We then sequenced 20 other candidate genes that resided in the minimal disease locus region, but did not find any disease-related mutations in them either. Finally, we conducted a genome-wide search for copy number variants using array CGH. To our delight, we identified duplicated regions in 6q27 in affected individuals from 4 of the 7 families. The duplicated regions in all 4 families contained only one known gene, the T gene. Our report of this finding was the first time that a copy number variant had been identified as the causative mutation in a cancer predisposition syndrome, and it helped stimulate investigations into the role of brachyury and biologically related molecular pathways in the etiology of chordoma and the development of molecularly targeted therapies. These are the 4 families that have the T gene duplication, or as we refer to it as T-dup+. And one thing you can notice about them is they all have at least 3 affected individuals with chordoma. These are the 3 families that didn't have a duplication in the T gene. And the striking thing about them is there's only 2 affected individuals in each family. Our clinical studies were based on evaluations of 3 of the 4 T duplication plus families and the 3 duplication minus families. We compared the familial chordoma cases with sporadic cases from NCI's Population-Based Cancer Registry, SEER, which stands for Surveillance, Epidemiology, and End Results. The T duplication plus cases differed from sporadic cases in SEER in 3 major areas. 93 of the T duplication plus cases presented with skull-based tumors versus 37% in SEER. The T duplication plus skull-based chordomas were diagnosed at a median age of 26 years versus 49 years in SEER. And only T duplication cases had multiple primary chordomas. We found 3 such individuals in our families. We could not assess the presence of multiple primary chordomas in the SEER data, but reports of multiple primary chordomas in the literature are in fact very rare. Finally, the T duplication minus cases differed from the sporadic cases in SEER only in one aspect, and that was that 83% of them presented with skull-based tumors versus 37% in SEER. These findings indicate that the diagnosis of chordoma at a young age, presentation with skull-based tumor, or the presence of multiple primary chordomas merits careful review of family history. If the family history is positive, then it's possible to screen at-risk relatives by MRI for early detection of chordoma. In addition, identification of a specific mutation in a family permits surveillance to be limited to mutation carriers. And finally, one should always consider imaging the entire neuroaxis if a patient is diagnosed with chordoma at an early age or as a blood relative of a chordoma patient. My NCI colleagues are continuing to investigate the genetics of chordoma in both familial and sporadic cases, as well as studying somatic changes in tumors. I want to thank the organizers of this symposium for inviting me to participate in it. It has given me the opportunity to complete a circle, to give the last presentation of my career at the place where it began and in honor of my mentor. I consider myself very fortunate to have had a career that has brought me great satisfaction on an almost daily basis, outstanding colleagues, and long-lasting friendships, some of which date back to my graduate school days. I know how fortunate I have been, and I attribute much of that great fortune to the years I spent in Larry's lab and here at the University of Washington. So thank you very much.